Good afternoon, and welcome to the PRISM e-Symposium. I am Leslie Porter-Cabell, and I will be serving as moderator for this session. Derek Sokolowski is my co-moderator.
Hello, everyone. This is Derek.
You are on the Toxicology concurrent session. This session will have two parts, one from 5 to 5:30 p.m. and one from 5:35 to 6 p.m. In this session, we will hear from students presenting on topics related to toxicology. If you have questions, please use the Q&A button below to write them and send them to the host. The host will select questions at the end of each presentation. Next slide, please. Our first presenter is Frank Gutierrez. The title of the presentation is Development of an Analytical Method for the Determination of Cannabis Exposure During Pregnancy. Thank you, Frank.
Next slide, please. Thank you all. In my presentation, I will give you some background of cannabis in the United States and why my project is concerned with it. I'll speak on our hypothesis and research approach and finish with the discussion of results and next steps. Next. Although cannabis is still illegal in the United States, many states have relaxed the criminal laws surrounding cannabis, and some have even gone as far as to legalize the drug. This has led to society becoming more accepting of cannabis. The problem with the normalization of cannabis is that some people who shouldn't be consuming the drug are. In this presentation, I will be focusing on a method to determine whether or not a pregnant woman has consumed cannabis by analyzing their placenta. Next. The problem with pregnant women consuming cannabis is that research has shown that this could have negative impacts on the development of the newborn. These side effects include lower birth weight, shorter body length, small for gestational age, and preterm birth. Additionally, prenatal exposure to marijuana has been linked to negative effects later in life, which include smoking marijuana at a younger age, following negative adult roles, and lower intelligence compared to others. Next. Although it would be much easier to simply ask a mother whether she had consumed cannabis during her pregnancy, these results would be biased to prevent legal action of the government upon her and her child. An objective, accurate, and precise analytical method is thus needed to determine whether or not cannabinoids, the molecules found in cannabis, are present in the mother. It has previously been found that cannabinoids can be found in the meconium, which is the first stool passed by the fetus, and the umbilical cord in mothers that have previously consumed cannabis. Our project hypothesized that cannabinoids would also be present in the placenta, and we began to develop a method to test this hypothesis. Next. Our objectives for this project were to develop a methodology which would quantify the 8 cannabinoids we were looking for in placenta. These 8 compounds are shown on screen, and we wanted to quantify them via liquid chromatography tandem mass spectrometry, abbreviated LC-MS. With this methodology, we would then test authentic placenta samples that are presumed to already have cannabis in them. Next. We took placenta samples that did not contain any cannabinoids to begin testing our method. The first step is to fortify them, which consists of adding the cannabinoids to the sample in known quantities. THC and its internal standard are shown here as an example. The internal standard serves as a reference to the analyte and allows us to analyze the ratio of cannabinoids to internal standard for quantification. The sample was first homogenized with methanol in a bead mill, then centrifuged. This removes the solid tissue and other contaminants from the supernatant, where the cannabinoids are. The supernatant is evaporated, leaving behind the cannabinoids and other molecules that weren't removed during centrifugation. A solution of formic acid, acetonitrile, and water is added to the molecule, and the sample was centrifugated again to further remove contaminants. This supernatant is then transferred to a strong cation solid phase column. This column functions by attracting positively charged cannabinoids to its negatively charged stationary phase. After the supernatant is diluted from the column is treated with different washing solutions to selectively remove some of the contaminants that may have adsorbed to the column. Finally, the cannabinoids are removed from the column using an elution solvent and are collected. The solution is evaporated, leaving behind the cannabinoids. A solution of formic acid, acetonitrile, and water is added to the cannabinoids, which is injected into the LC-MS. Next. Inside the liquid chromatograph, the cannabinoids are separated in the column due to trackings to the packing and to the mobile phase. Once the cannabinoids reach the end of the column, they are injected into the mass spectrometer, where they are ionized, fragmented, and put into the transducer, which detects the signal. The output is a chromatogram that gives the retention time of the analyte with how much signal it created at the transducer. This signal is correlated to the concentration of the analyte. The chromatograms shown are for each of the cannabinoids we analyzed. Next. Once a method that yielded accurate results was established, the method was put through a validation step by the scientific working group of forensic toxicologists. The results are shown on the right. Additionally, authentic placenta samples that contained cannabinoids were tested for cannabis using our method by creating a standard curve, which is on the left, where we know how much of a cannabinoid is in our samples, and then measuring the response obtained from the authentic samples, we could accurately determine how much cannabis was in an authentic placenta. The data we found also correlated with the cannabinoid concentration determined in other matrices from the same person. Next. Our work shows that the LC-MS is sensitive enough to analyze cannabinoids in placenta. Next steps for this project include testing the rest of the authentic samples and finishing the validation process of our method. Next. The authors would like to take the opportunity to thank PRISM office for their support. This research is being done in collaboration with La Universidad Santiago de Compostela in Spain, and we further appreciate the placenta samples they and El Complejo Hospitalario Universitario obtained for us. Thank you all for listening.
Thank you, Frank. We have one question for you. Can this method be adapted to detect other drugs?
Likely not. These drugs are more of an acidic nature, and other drugs could be basic or neutral, depending on the pH of the solution. And we would have to modify this this procedure to detect these drugs and to quantify them. And this would not be the same for other matrices as well. Like we would have to modify this protocol to be able to quantify the same cannabinoids in different matrices.
Thank you, Frank. Okay, our next presenter is Atera. The title of the presentation, The Role of Microorganisms on Mercury Flux from Natural Soils. Atera?
Thank you, everybody, for being here. Next. We all know that mercury is an unsafe chemical. It is a carcinogen, it is a mutagen, and can cause nervous toxicity. Now, mercury cycling can occur in the atmosphere during natural saturation processes such as rain. So during rain, it brings mercury ions from the atmosphere and deposits in the soil. Now, once the mercury ions are in the soil, when it gets exposed to abiotic factors such as UV light, heat, and humic acid, the mercury ion then gets reduced to mercury element. After this reduction mechanism, mercury element then gets emitted back into the atmosphere. Next. Now, in a research conducted by two scientists named Das and Das, it was found that some gram-negative bacteria can actually turn ionic mercury to elemental mercury on its own. Now, based on the preliminary research, we had hypothesized that the reduction of mercury can occur through gram-negative bacteria in darkness while having other abiotic factors constant. Next. Now, the way that we went about this research is that at first we had collected and prepared the soil samples, and after that we dried the soil, and then we measured the mercury levels, the initial mercury levels, using this instrument called DMA-80. After that, we saturated the soil, and then we measured mercury emissions using this instrument called Techrin 2537A. Next. Now, in this part of our research, as Elvin's research will show, autoclaved soil did not show any mercury emissions. Here in blue, we have our control soil sample, and in yellow, we have our experimental soil sample. We see continuous mercury emissions in the control soil sample, whereas we see significantly decreased amount of mercury emissions in the experimental soil sample. Next. Here, we found that mercury emissions increase with bacterial food. In blue, we have the control soil sample, whereas in orange, we have our experimental sample. Here, the control was saturated with sodium chloride solution, and we see a continuous emission of mercury there. In the experimental sample, the first and the fifth peak had presence of bacterial food, LB broth, and the second, third, and fourth peak did not have any bacterial food. Here we see a sharp increase in the first and fifth peak, whereas we don't see a significant change in the second, third, and fourth peak. Next. Now, sorry, can you, go back. Yeah, here. Presence of antibiotics does not show any mercury flux. Here in orange, we have our control, and in blue, we have our experimental. Now, the orange did not have any antibiotics in it, whereas the blue peak shows soil samples that did contain antibiotics. Now, when we compare these two soil samples, we don't see any significant changes between them. Next. Now, from this research, we can conclude that autoclaving, or essentially sterilizing our soil, were found to significantly decrease mercury flux, whereas stimulation using bacterial food were found to increase mercury flux. And also from the antibiotics results shown in the previous slides, we also deduced that these bacteria are antibiotic resistant due to no significant changes being observed between the samples. This result provided us enough evidence to prove our hypothesis that microorganisms, specifically gram-negative bacteria, have been shown to be active participants in the reduction mechanism of mercury in natural soils. Next. Thank you to PRISM, and thank you to Dr. Carpi, Darian, and Hamid Patel for helping me in making this presentation. Thanks, everybody.
Thank you, Atera. We have a question. Do you expect results to be different with the gram-positive bacteria? Why or why not?
We think the results would be different with gram-positive bacteria because when we sterilized the soil, there was significant decrease in bacterial activity, right? And when we added the antibiotics, the mercury flux went down. So that means that the bacteria had to be gram-negative.
Okay. Thank you, Atera. Our next presenter is Elvin Colón. The title of the presentation, The Effect of Sterilization on the Reduction and Emission of Mercury in Soil. Thank you, Elvin.
Thank you. Hi, mercury contamination is global and affects many bodies of water, causing harmful pollution. This is because mercury emissions generally disperse widely in the atmosphere before being deposited onto the Earth's surface. Exposure to this toxic element threatens our health while also causing damage in the ecosystem. So what is mercury, you may ask? Mercury is a chemical element naturally found on Earth's crust. High or continuing exposure can cause damage to the nervous system by changing the synthesis and the release of neurotransmitters like dopamine. This damage leads to both emotional disruption and behavioral changes. Mercury in the atmosphere is transported over a range of distances before it is deposited by rain. When released, the element undergoes reduction from Hg2 into Hg0. It then pollutes land and sea, thus affecting living organisms. Next. Besides focusing on water in the past, we studied the effects of abiotic factors such as light and temperature that showed increases in mercury flux, which indicates mercury reduction. However, when the effect of water was studied in the absence of any other known abiotic factor, we had similar results. The first approach taken was using the soluble components of the soil to obtain a soil leachate and saturating our soil samples with the soil leachate to see any effects. However, no significant change in mercury fluxes were observed. The second approach was where we suggested it may be due to the presence of microbial communities that may facilitate an Hg reduction. So in this study, we attempt to sterilize the soil to take out the biological factors in our sample. Next. A batch of soil is taken into the lab, shifted to homogenize, and separated into two samples. One sample is sterilized twice using an autoclave, while the other sample acts as a control. The autoclave is a sterilization method that uses heat and pressure to kill off bacteria. The direct mercury analyzer will analyze initial total mercury concentration before and after the autoclave step. Samples are spread in a 1 centimeter deep circle on a Teflon-lined metal tray, then transferred into flux chambers. The Techran will measure gaseous mercury concentration inside the chamber, and our samples are watered at 40% of their given weight, which is 25.8 grams, and given 4 days to fully dry in between watering. Next. The 4 water additions can be seen in the graph as peaks in mercury flux. Results show that the emissions of mercury are significantly lower in autoclave soil, shown in yellow, compared to that of the controlled soil in blue. The trend in our control sample is what we expect from previous experiments conducted. We can see that our autoclave sample showed very minimal amounts of mercury being emitted, with little change throughout subsequent watering. Next. Here, we see a better description of what's happening. This is an integration of the previous graph over equal time intervals per peak. Our autoclave sample peaked at its highest during the 4th watering. We also see a trend of mercury emissions in our autoclave sample, increasing during the end of the experiment. The controlled sample peaked at its 2nd watering, then gradually decreases in emissions after the final half of the experiment. Next. To summarize, we can infer that the autoclaving procedure killed enough microorganisms to affect the way emissions of mercury would respond otherwise to multiple water additions, and it provides strong evidence that microorganisms seem to play a large role in mercury emissions on wet soil. Next. Further experiments will be made to determine whether similar patterns will continue when different types of soil collected from different locations and sterilization techniques are used. The goal of this is to uncover similar behaviors that may rise and to make more general claims as to the mechanism in play. Next. I would like to thank Dr. Anthony Carpi, Darian Menard, and Hamal Patel for helping me create this poster. Thank you.
Thank you, Elvin. You have a question. Why did you autoclave the sample twice?
Well, when autoclaving the sample once, we've experienced the emissions of mercury continuously rising. So there was still microbial communities present in our sample.
Okay, thank you, Elvin.
Thank you.
Our next presenter is Rader Rodriguez. The title of the presentation, Analytical Determination of Methylone and Metabolites in Brain Tissue by LCMS-MS. Thank you, Rader.
Hello, next. Hello, everyone. This is an overview of my presentation, which includes a little background on my research project, the research approach that I use, data, discussion, and future directions. Next. Methylone is a psychoactive drug if abused that belongs to the synthetic cannabinoid family. It is an illegal drug better known on the streets as bath salts, and during the last decade has been targeted as a public health threat due to its high addictive properties. Methylone has been known for having amphetamine-like characteristics in terms of its mechanism of action. However, not much is known about its pharmacological effects in the brain. So for this reason, scientists have been working on the development of efficient analytical techniques with the purpose of using them as a tool to study the pharmacokinetics and pharmacodynamics of this drug in diverse biological specimens. Next. For this project specifically, my goal has been to develop and validate a reliable analytical method based on LCMS-MS, which is a very known analytical technique for the quantitative analysis for multiple compounds. So this method not only seeks to identify and quantify methylone and metabolites in rat brain tissue, but also it helps to be used as a tool to study the pharmacological behavior of this drug in the brain. Next. So the way I developed this method was basically by following a series of parameters for method development that includes sample preparation, techniques, LCMS analysis, and validation. Next. So sample preparation consisted of a series of procedures for drug extraction from the brain tissue. This included homogenization of methylone dose rat brain in buffer solution, beta-glucuronidase hydrolysis, protein precipitation, evaporation, reconstitution of samples, and filter virus for analysis, which was done by using LCMS-MS instrument equipped with a synergy polar RP column and a triple quadruple mass spectrometer. After LCMS-MS analysis, different experiments were performed during a process of validation, and the validation process estimates the efficacy and reliability of the method, but also provides any method limitations under normal conditions. Next. So after analyzing the samples by LCMS-MS, we obtained results that indicated the distribution of ions by mass, as well as the retention time of each analyte. which is basically the time from injection to detection of the drug. This was translated into chromatograms that show different peaks for each analyte, and these peaks represent the retention time versus the intensity of abundance of each drug detected. Next. The results for method validation were evaluated by following parameters established by a scientific working group for forensic toxicology. In the method validation, we tested for calibration, bias, imprecision, matrix effect, extraction, and process efficiency. So most parameters presented a coefficient of variation within 20% according to the guidelines for method validation, which makes this method reliable in terms of sustainability. So in addition to that, all experiments for validation were performed using quality controls to preserve the integrity of the method. Next. A total of 10 authentic brain samples were tested for methylone and metabolites. The concentration range for the analyzed in authentic brain samples was from 5 to 2,250 nanograms per gram. Analyzed that registered the highest reliable concentration were methylone, MDC, and HMMC, while HMC was not detected during the analysis. So multiple reaction monitoring chromatograms were obtained to observe the distribution of volume by mass, intensity of the analyte versus retention time, and concentration. Next. So based on the results that I got and the guidelines from the SWG Tox, this analytical method was proven to be sustainable and reliable for the identification and quantitative analysis of methylone and its metabolites. Future directions include the incorporation of this method for the quantitative analysis of methylone in forensic and clinical settings, as well as the use of this tool to study pharmacological effects of methylone in the brain. Next. Finally, I would like to thank the whole Dr. Concheiro-Guisan lab for all the support and all the lab members that were directly involved in this project, and of course the institutions that made this event possible, such as CSF and NSF and PRISM staff.
Thank you, Rader. You have one question. Could this method be adopted for other clinical applications?
Well, based on the literature has shown different methods for methylone based on LCMS. However, this method was created specifically very unique and designed specifically for identification and quantification of this drug in rat brain. But in order to be useful for other biological specimens and other techniques, it needs to be reevaluated for the whole method based on the type of biological sample, such as urine or blood. And we have to reconsider all the parameters again, LCMS and sample preparation.
Thank you, Rader.
You're welcome.
Our next presentation is Keiann Simon. The title of the presentation, Method Development for the Quantification and Qualification of Nicotine Cannabinoids and Vitamin E and e-cigarette liquids. Thank you, Keiann.
Thank you, Leslie. Next slide, please. Now, many of you have heard about or partake in this new vaping trend that mostly, but not limited to, younger people have been indulging in. Generally, e-cigarettes were first used to facilitate nicotine products, but had quickly graduated to recreational drugs, vitamins, and many different types of substances. Due to this next step in industry, there lay a greater need for the investigation of the exact qualitative and quantitative information about these substances in the e-cigarette liquids. Next slide, please. The clinical effects of nicotine, cannabinoids, and vitamin E intoxication through e-liquid ingestion remains uncertain. However, the biggest health concern we see from case studies and scientific research is lung degeneration and developmental issues in fetuses and young adults. Moreover, they found that children and adults were poisoned when exposed to the e-cigarette liquids through swallowing, breathing, or absorption through skin or eyes. Next slide, please. Due to this knowledge gap and delirious effects that are progressively popping up in society, in vaping devices, our research aims to develop a methodology to detect the tobacco derivatives such as nicotine, cannabinoids, and additives such as vitamin E within e-cigarette liquids. Next slide. Our research approach can be divided into 3 steps. The first step is to consult the literature to see what has been done and what instruments are appropriate for the substances we are trying to screen for. The second stage implements the validation of previous literature results and developing and optimizing a new protocol in which parameters are going to be manipulated to suit the samples we will be evaluating. This will be done by running a series of derivative standards of nicotine, cannabinoids, and tocopherol, which is vitamin E, to determine the best instrument and ultimately the limit of detection range for each substance. The final step will include the application of the protocol developed in the first stage to authentic samples of the protocol of the vaping liquids to accurately detect and quantify the substances. Next slide, please. From the literature, we decided to use these two instruments to optimize nicotine, cannabinoids, and vitamin E. On the left, we see the liquid chromatography tandem mass spec instrument, the LC-MS-MS, and on the right, we see the gas chromatography mass spec instrument. We have started, however, to optimize standards on the LC-MS-MS, and I will be talking a little bit about what some of our results look like. Next slide. Now, MRM transitions are multiple reaction monitoring, and it's the most common method for quantitation of analytes by the LC-MS-MS. In MRM, ions are selected to make it through the first quadrupole and into the collision cell where they are fragmented. These ions are referred to as the precursor, quasi-molecular, or parent ion. Certain fragment ions referred to as product or daughter ions are then selected to make it through the second quadrupole. The transition from precursor or parent ion to product or daughter ion is referred to as an ion transition. Next slide, please. An example of the product ion scan of one of our standards, delta-9-THC, can be seen here, where we obtain the MRM transitions. We see in the red, the quasi-molecular ion at 315, and in green, the two prominent fragments at 193 and 123. Next slide. This is a summary of the mass spec optimization of the standards we have tried so far in the form by using LC-MS-MS. There are 9 cannabinoid derivatives represented in green and 3 vitamin E derivatives represented in blue. From the ion scan, using the example of delta-9-THC, we see therefore, as previously mentioned, the parent ion or precursor ion at 315 and the most intense ions at 193 and 123. This gives us 2 ion transitions for delta-9-THC. Next slide, please. For our future work, we will continue our optimization on the LC-MS-MS with our nicotine, tocopherol, and cannabinoids. And we will also try our optimization on the GC-MS. We will acquire our authentic samples and do various extractions and method validation. Next slide, please. I would like to thank the Concheiro-Guisan lab and PRISM for making this presentation and this lab work possible. Thank you.
Thank you, Keiann. You have a question. How do you use the MRM transition fragments to figure out what is in your authentic samples?
Okay, so the MRM transitions are analogous to a fingerprint in my terms. And you're creating essentially a database in which you can run your samples and match up to your optimization method and to see what compounds you have in your liquid or your authentic samples.
Thank you very much, Keiann. With that, we conclude our session. I would like to thank our presenters for their work and ask everyone in our audience to send them a big round of applause. Our next session on toxicology starts at 5:35 in the same room. You can stay here until it starts, but if you'd like to check out a different session, you can close this window and go to our website, www.jjay.cuny.edu/prism, and follow the link to join a different session.